Okay, so week one, we're going to be talking about anatomical terminology and microscopes. So first, here are our different objectives. So we're going to talk about anatomical terminology. We will become familiar with just a brief overview of what anatomy and physiology even means. We'll look at some organ systems. We will also become familiar with the microscope, the different parts of it and how it works. And then finally, just how to care for the microscope, and just a little bit of math, which won't be so bad. So first off, um, again, for people that didn't have class this week, I always make sure that everyone pays really close attention to your red terms, your underlined terms, and your bolded terms. That is what we are most likely going to ask you questions about. So make sure we know that those words really well. So first up, anatomy. Anatomy is basically asking you, what is this structure? So if I asked, what is this structure right here? You could say that that's the arm, that is anatomy. Physiology is, how do these structures work? What do they do and how do they do it, right? So what is their function? Anatomical position, you can see in the picture to the bottom right, this is a man standing in anatomical position. So your feet pointed forwards and your hands and palms down to your sides also pointing forward. This is really important because when you're talking about anatomy, this will keep everything the same across the board, so everyone will understand what you're talking about. And as future healthcare providers, an important aspect about this is understanding your left from your right. In anatomy, left and right, you always refer to the patient's sides and not your own. For example, if you guys are watching on the video right now, I can raise this hand, and to you, it may look like it's my left side, but it's actually the right. So when you talk about left versus right, it's always the patient's point of view. Next, some directional terms you see at the bottom. This is basically scientists and healthcare providers' way of describing different directions in the body. It's kind of like their own little language, right? So beginning at the middle, actually, we'll talk about superior and inferior. So superior is, it means closer to the head, so further up. And then inferior means closer to the feet, which is down. So superior and inferior are your up and down. Next, you have medial versus lateral. Medial means something's in the middle. So medial for me would be right here. And lateral means away from the midline, or if this is my middle, would be out this way, or this way is lateral. So medial means middle, and lateral means away from the midline or to either side. Next, anterior versus posterior. Anterior just means your front, or ventral is the same thing, and posterior and dorsal mean your back. Again, the model in the front, or I'm sorry, in the bottom right of our slide, you can see that his front side, that would be anterior, and then his back would be posterior. Now, proximal versus distal is a little bit more confusing. This proximal right here means you're in close proximity, so something is close, and distal means further away. But these two words are always in reference to a point that you're talking about. The example would be if I'm referring to my chest or my torso. That's a lot of that. That tends to be the reference point a lot of times. So proximal means something is closer to the reference point, and distal means it's further away. Again, the example being if my chest is the reference point and we're talking about my arm right here, we could say that my elbow would be more proximal to my chest, and then my wrist would be more distal. It's a little bit further away to the reference point itself. Over here on the right-hand side, we have our different planes of section, and this is just a way to divide the body up. So you can see they're all nice and color-coded. So the green cut is the frontal or coronal plane, and that will divide you between your anterior, your front, and posterior, which is your back. Next, we have our sagittal plane, which will split you right down the middle and divide you into left and right. And then transverse planes of section will cut you right down the middle and divide you into your superior and inferior aspects. Transverse planes are used a lot in like CT scans. If anybody's ever seen one of those, it looks like a circle or like kind of like an oblong shape. There's a bunch of different like spots in it and stuff, and nobody ever knows what it means besides radiologists. But that is a transverse view of your body. So body regions and landmarks, you can see that this whole thing is in red. In class, we don't talk about each one of these individually, but you will need to know them. So make sure you guys star and highlight this slide right here. It's very, very important. Just for time's sake, we will not explain each one of these words to you, but you do need to know all of these words on this slide right here. This is just a way to use medical terminology to describe an area in the body. And you will use this as an aspiring healthcare provider. You'll use these words every single day. So make sure we know all of these words on this slide. Next, the abdominal regions. You can see in the picture that we have off here to the right, the abdominal region in the left is divided into 9 different regions, but the one on the right has 4 quadrants. These basically, are just 2 ways of describing the abdominal regions. They're both very valid. The one on the left is used more for PhDs, like scientists, and the one on the right is used a lot more in healthcare. However, we want you guys to be familiar with both. As you can see down at the very bottom, we have a few words to help you kind of figure out what you're trying to remember and memorize here. So you can see that hypo means below, and then epi means above. So an example of hypo would be if you are hypotensive, that means you have low blood pressure. The opposite of that being hypertensive, you have too high of a blood pressure. So hypo means below. Epi is another way to say above. And then chondriac refers to cartilage. So let's look at the left hypochondriac region. You can see again that this is left, even though the way that we're looking at it, it seems like it's on the right. But remember, it's the patient's sides that matter. So this is the patient's left, and it's hypochondriac, which means below cartilage. And you can see that in this picture right here, that left hypochondriac region is right below those lower ribs, which have a lot of cartilage on them. And we'll explain a little bit more about that when we get to the bones in a few weeks. But you will want to know all of these different regions and their names in the order that they are in, as well as the quadrants. And you want to know one organ that you can find in each region. And you can see them all listed over here on the far left-hand side. For example, the right hypochondriac region, so right below the cartilage, you have the liver, large intestine, and gallbladder. So again, memorize all of these sections. I would recommend drawing a big tic-tac-toe board on like a whiteboard in the library, writing out all of these different sections and these regions, and write one organ that you can find in each region, because that's how we will ask you questions on the practical. Next, some body cavities. A funny way that I use to describe what body cavities are is that I always say that the body is kind of like a refrigerator. So imagine opening up a refrigerator and you see all of the different shelves, and you have the 2 drawers where your mom always puts all the lunch meat, and then at the very bottom you have some more shelves, and then you have like those big buckets that you can pull out and put like fruits and vegetables and stuff in there. The body also has its own cavities that the organs will sit inside of. So starting at the top right here, we have the thoracic cavity, which you can see in purple, and you have 2 pleural cavities, which is where there are 2 kind of just normal cavities that each lung will sit in. So those are your pleural cavities. Next, you have your mediastinum, which houses your esophagus and trachea. You can see that in orange. And And again, for recording's sake, I'm not sure if you guys can see my cursor, but I'll try to be very descriptive of what I'm talking about if you cannot see my cursor. Next, we have the pericardial cavity, which is in the right side of the picture, this blue structure right in the middle that houses your heart. Continuing on, we have the abdominopelvic cavity, which you can see in the bottom right, and that's kind of a combination of the abdominal cavity and the pelvic cavity. So even though those are two separate cavities that house their own structures, you can still refer to them as one, being the abdominopelvic cavity. Finally, on this slide, we have membrane layers. Jumping down to the bottom, we have visceral layers, which lie directly against the organ, and parietal layers, which lie against the wall of the body. Now, the reason for these layers is that they separate organs from each other, okay? And these membranes are serous, which means they secrete fluid. So you'll basically be having, if there's an organ over here and an organ here, you have some layers and there will be fluid in between them. And the reason for this is it helps to protect the organ, and it would also reduce friction. So friction would be bad in the body because if two organs were rubbing up against each other, it could damage them, and it could also create, it could increase your body heat. So you know when you're outside and it's really cold and you start to do this, right? You're rubbing your hands together. That's creating friction, which creates heat. So if you're, let's say you went for a jog and all of your organs inside of your body were rubbing together and creating all this heat, that would be really, really bad. So we have these membrane layers that not only help you to divide between the different organs to keep them separated along with these cavities, but they secrete this fluid to reduce friction and to have some form of protection. Next, we have our levels of organization. You can see over here in anatomy and physiology, we're going to talk a little bit about all of these things. But the atomic and molecular level, you'll focus solely on in chemistry or biochemistry, as you can see here. The study of cells, which we'll talk about next week, is called cytology. Cyto meaning cell, or just cyte meaning cell, and then ology meaning the study of, so the study of cells. Histology is the study of tissues. And what you want to know on this slide are these levels of organization in order, okay? So the smallest thing that we have would be an atom. So example is one carbon atom. A bunch of atoms combined together create molecules. Lots of molecules together can form a cell. A cell is made of a bunch of different molecules. A whole bunch of cells then combined make up tissues. Then tissues, when they come together, they form organs. A bunch of different organs together create a system. And then us as humans, we are built up of a bunch of systems. Okay, so you want to remember each of these terms and know them in order as well. You'll also want to know all 11 organ systems. So here are your 11 organ systems, and for the quizzes and the practical, you want to know each one and one of the major structures or organs that's found in this system. So first we have the muscular and skeletal systems. Your major organs in them are muscles and bones, right? Pretty straightforward so far. Next we have the nervous system, which is composed of your brain and spinal cord and all of your nerves. And this is for sending information and gathering information in from the outside world. It's kind of like the computer inside of your body, which is really cool. And we'll talk about this for the third practical in Bio 214. Next is endocrine. Your major organs here are all of your glands, and that will be a big topic when you guys get to 216. Next is the cardiovascular system. This is made up of the heart, the blood, and the blood vessels. and their function is to transport nutrients and oxygen to your cells, and then to take wastes away. That's a 216 topic as well, which is really fun to dive into once you get there. Now you have the integumentary system, which means your skin. So your skin is your major organ, and we will talk about that in 2 weeks. Now we have the lymphatic system. This is like your immune system, and your major organs can be the lymph nodes, lymphatic vessels, your spleen, thymus, and tonsils. And the respiratory system over here obviously is used for breathing, and your major organ for that is the lungs. These two are also 216 topics. Then you have the digestive and urinary system. The digestive system is to break down the nutrients that you eat and kind of metabolize them. Major organs include the stomach and your intestines. The urinary system, the major organ are the kidneys, and those guys filter your blood to remove nitrogenous wastes. These two are also 216 topics. And again, this may seem like a lot of information for you guys, but all we really need to know here are the 11 organ systems, what are they, and one organ that you can find in that system. And then finally, we have our male and female reproductive systems. That is also a 216 topic. So everything that I mentioned is in 216. You guys will dive in much deeper once we get there next semester. So that was the end of chapter 1. Now we're going to move into chapter 2, which is about microscopes. So first right here we have this red term, which means it's important, called resolution. So resolution is the amount of magnification required to distinguish two entities as two distinct points as opposed to just one point. Basically, all this mumbo jumbo means is that pretend like you are on top of snowflakes up on Liberty Mountain, and you're looking out at the Blue Ridge Mountains, okay? And you are looking at a mountain, and it looks like a huge, huge mountain because you're really, really far away. Then you hop in the car and you drive even closer to the mountains. And as you get closer to those mountains, those same ones, what you originally thought was just one huge mountain may actually be a few different smaller mountains that just happen to look like one big mountain. But as you got closer, you were able to tell the difference, right? That is what resolution is. In terms of the microscope, instead of looking at something that's massive like a mountain, we're looking at something really, really tiny like cells. So this whole thing right here where it says it's the amount of magnification required to distinguish two entities as two different things instead of one. If you're looking at something in the microscope and it looks like one big blob, and you increase the resolution or you increase the magnification so you get closer to it, you may be able to tell that that thing that looked like one thing may actually be two separate smaller things, right? So that's what that's talking about. Next, your human eye can see fingerprints. So if you look at your finger, you can see your fingerprints, but you can't see cells. But the light microscope can see cells. However, it's not strong enough to see organelles. To be able to see even smaller than that, you would need like an electron microscope, which we don't even deal with. These metric measurements over here, I will want you guys to be familiar with. And just a forewarning, on the practical, you will not have to do any math. So we're going to talk about some math, but we will not have to solve any equations or anything like that on the practical. This, however, you do need to know. You will want to look at all these pictures of the microscope and know all of the different parts, because that will 100% be on your practical. So make sure this picture off to the right is really, really good. So be able to label the microscope and know all of its different parts. And at the bottom here, here's our first tiny bit of math. Something that you may be asked maybe this week on the quiz, and it's just good for general knowledge and understanding, is total magnification. Okay, so this is like the strength of the magnification that you have when you look through the microscope. So as you can see right here, we have this thing called the ocular lens, which is the eyepieces that you look in when you're looking into the microscope. Inside of there, we have a lens that has 10 times the magnification. So an example is, let's say I had a quarter in my hand and I was only looking through an ocular lens. If I looked through here at that quarter, the quarter would look like it was 10 times bigger than it actually was. Following me so far? Now, you also have these objective lenses, which are the ones that you rotate around when you're using the microscope. Again, I'm not sure if you can see my cursor, but you can look at these pictures, find where it says objective lens, and that's what we're talking about right here. You have three different ones. You have scanning power, low, and high power. Scanning by itself magnifies things by 4. Low power magnifies things by 10, and high power by 40, okay? Now with this total magnification, it's asking you, how much am I magnifying something in total? Because remember, you are looking through the ocular lens and the objective lens at the same time. So to find the total magnification, all you do is multiply your ocular lens by the objective lens. And so you can see those total magnifications over here. So scanning's total magnification will always be 40. Low power's total magnification will always be 100. And high power's total magnification will always be 400. Now here's where I tend to lose some people. Field of view. Field of view is a very simple concept that people make way harder than it needs to be, okay? Field of view is literally the distance that you are looking at when you look through a microscope, okay? So if you're looking through a microscope, you are measuring the distance that you are looking at. And since we're looking through a microscope, remember, the things that we will want to look at are very, very small. In lab, if you guys were not there this week, one of the activities is to put a ruler, like the one in this picture off to the right, put a ruler under the microscope and look at the different lines under scanning and low power. And you would see an image similar to the one in this picture where you can see the different tick marks, right? So measuring the field of view is the same thing as if I were to pull out a measuring tape and measure the size of my computer monitor right here. All you're doing is measuring the distance that you can see under the microscope, okay? So to calculate to calculate the field of view, you will only have to do math if you want to find the field of view under high power. Now here's why. This image off to the right where you can see these 4 different millimeters could be under scanning power, let's say, okay? Now, if this is under scanning power, it's easy to measure this because we can still see all 4 lines. Now, which would make the field of view 4 millimeters, okay? If we were to increase the magnification to low power, so we're going from a total of 40 total magnification to 100, so we're increasing magnification, we would be getting closer to the ruler, right? If that happened, we might only be able to see like 2 millimeters instead of 4 because we're getting closer to it, right? Now, if we were then to move the objective lens to high power, we would be going from 100 total magnification to 400. We're getting so much closer to the object. And you would notice that when you looked in there, you wouldn't be able to see anything. And that is the reason we have to do this math that you see at the very bottom. We have to do the math because you can no longer measure the field of view with just your eye, okay? So again, on scanning power, if our field of view was 4 millimeters, you increase it to low power, maybe you could see about 2-ish. Increasing it to high power, you would not see any more tick marks because you will be focused in between two of those lines. So I used this example in class. If here's my 4 lines, and I'm on scanning power, I can see all 4 lines. If I were to zoom in like this and get closer, I could maybe only see 2 of them, right? Then if I were to zoom in one more time to high power, I would be looking right in between 2 of those tick marks. And since I can't see any tick marks, I can't measure it anymore. And now we will explain the equation of how to find that. So you can see that at the very bottom left hand of this PowerPoint. And it reads the field of view on high power multiplied by the total magnification on high power is equal to the field of view on low power times the magnification on low power. Okay, so this field of view on high power, remember, is what we're looking for. So that's kind of like our variable. You know, in high school algebra, when you guys took that, you solve something for x. This is our x. This is what we're solving for. Now, the magnification on high power, again, the total magnification is always 400 for high power. Remember, the 10 in the objective lens multiplied by the 40 in the, the 10 in the ocular lens multiplied by the 40 in the objective lens. Now, the field of view for low power, again, you can just follow this arrow. I just used this example right here because we don't know what objective we were actually looking at this. So we'll say that the field of view on low power that you guys would have measured would be 4 millimeters. So we put 4 millimeters for that. And the magnification on low power is always 100. Remember, 10 times 10 is 100. Multiply everything out and you'll get 400 is equal, or 400 FOV on high power is equal to 400 on the other side. Divide it all out and you get field of view on high power is equal to 1 millimeter. Remember, that's exactly what I just explained to you guys. On scan, on high power, you'd only be looking at 1 millimeter of distance. Again, don't worry about that so much. We really want you guys to just understand this concept, but you will not have to solve this on any practicals. Okay, this has been getting a lot of questions from people, so I will explain this to the best of my abilities for you all. Especially for those watching that are in quarantine, I'll explain this all to you right now. We have 2 main resources in this course that are required for you guys to buy. The first is a subscription from xatomy.com. You can see that written right here, xatomy.com. This is our lab textbook, and it also has, it will give you access to the lab activities and the quizzes, okay? So this is required 150%. The lab exercises, if you were in class, you will have done those. For those of you that may be in quarantine right now or not able to make it to class, once you can come back, we will make up these activities for you guys and with you so you can see all this fun stuff that we get to do in class. And each lab exercise will be worth 25 points. For those of you that are taking the course in class right now, after you finish the exercises and the activities for that day, your GSA will come around and we will get your grade right then and there in class. For all of these assignments, Canvas might tell you that they're due on Sunday night at midnight. That is not true. Everything that involves an assignment in this class will always be done in the classroom. Nothing will be due at Sunday night at 11:59. Canvas just puts that there automatically, okay? So don't worry about what Canvas tells you, worry about what your GSAs tell you. So again, lab exercises you do every single day in class when you come there. Next, quiz number 1 will be taken at the beginning of class next week, and that will cover what we just talked about right now. So review your PowerPoint and read through the chapters 1 and 2 on xatomy.com and shoot us any emails that you guys have. Lastly, we have a new book this year that's called the 214 Anatomy Atlas that's written by Dr. Blaze. This is what we will use for homeworks, and it will encompass a total of 50 points in the course, okay? This week, the homework in that book is pages 159 through 161, and it will cover the microscope and some of the anatomical terminology that we talked about. Now, this book has been giving people issues this week, and you will need to go physically to the bookstore and ask to buy a copy. For some reason, if you try to order it online, it's saying that it's back ordered. So do your best to get this book. If we're still having issues with it, or if we're having issues with people getting the book, we will have grace for everybody. So don't freak out. Don't email your GSA 9,000 times telling them that you can't get this book and that your life is over. We'll all get one eventually. So don't stress about that. If you have it, we'll record your grades as we go, and the pages 1